Having me here to give this presentation to you today. This is the title that I'll be presenting to you. I gave the same talk at a conference on plant cell walls earlier in July in Palm Cove on potato cell wall structure and starch analyses in cultivars with different digestibility, but I've sort of catered this a little bit, like Barbara said, to give you an overview on where I actually come from. So I'm currently a postdoc at a company in New Zealand called Plant&Food Research, and the company has many sites spread throughout New Zealand. It's got in total about 750 employees. The headquarters are located in Auckland up here, which is where I did my PhD and used to live, but I've now relocated and started my postdoc in Palmerston North, which is this little city down here. So the way Plant&Food Research operates is the sites are mainly focused around the crops that we research in the area. So for example, in Tauranga and Te Puke, we grow a lot of kiwi fruit in this region, so we focus a lot of the research on kiwi fruit here. In Napier and Hastings down on the east coast over here, we grow a lot of apples and summer fruit, so most of the research is pip fruit based in this region. In Palmerston North, where I work, there is quite a focus on food nutrition, health and wellness, those sorts of things. In Nelson up here, there is a site that does seafood research. And in Christchurch, this region grows a lot of lovely wine, so there is a lot of wine and soil research down here. And Clyde, down at the bottom, there's a lot of stone fruit, so apricots and things like that. So basically, Plant&Food Research is a CRI, Crown Research Institute. And a lot of our research is focused around crops that we can grow and can grow for export purposes, because New Zealand's a small island country, so we rely quite heavily on export. So that's the main reason a lot of our research is focused on these sorts of crops. Unlike here in Australia, where a lot of the work that you do might be on maize, barley, sorghum, things like that, but we can't grow stuff like that down there. It's just too cold. So this is basically what we do. Also, every time I present at a conference, every time I say I come from New Zealand, people say, Lord of the Rings, and I go, okay, yeah, I'll put it on the map. This is where it was filmed. This is what it's famous for. We're all quite proud of it down there. So, some of the products from Plant&Food Research, which you might or might not be familiar with, obviously kiwi fruit, which is one of the most popular ones. We have many different varieties that we grow, mostly for export. But some others that you might have heard of as well, like the jazz apples, came from our apple breeding program, as well as the envy. I don't know if you get them here yet in Australia. And more recently, we're working on things like red-fleshed apples, but in its current state, it's still in its transgenic form, so it's not commercially available yet. We also release products in the vital vegetables lines. I think I saw this one marketed in Australia, the booster broccoli. It's enriched with glucosinolates. So yeah, just some of the products that the company makes. And just a bit on the people that I work with. I actually come from two groups. So I'm split between the food and nutrition group, which was formerly led by Juliet Ansell, and also the post-harvest fresh fruit group, which I work with Dave Brummel up there. And Barbara over here knows Chrissy. Chrissy leads the animal and clinical trials that we do. So unlike you guys over here, you might do more animal trials than us. We do it on a small scale, so we just have mice and rat trials, unlike your pig facilities over here. And we have a bunch of scientists that also have, I think, a mutual interest in Mike and Barbara's group in terms of research focus. We do quite a bit on gut microbiota, fermentation, digestion, things like that. So very similar to work that Mike and Barbara do over here. I work with John Monroe and Simon Mishra. They have an interest in dietary fiber and digestion. The groups are actually quite small. The site that I work at, there is only about 100 people. So when I first came to UQ, I was really overwhelmed because the campus was huge, the department was big. And Mike got me to present this talk at his department. And he said, don't worry, it's a small group. And I showed them and they were like, so many people. And I was like, oh, you lied. Anyway, because, you know, small to me just says small. And there were so many people there. And over here in this group, I work with Dave Brummel and Erin O'Donoghue. They are plant cell wall scientists. And the groups are quite small. This one's got 4 technicians, 3 PhDs, and half a postdoc. This is me, by the way, the half a postdoc. 3 technicians over here. I also come from this virtual cell wall group. This is basically made up of individual scientists that belong to different groups, but they have a common interest in plant cell wall studies. So we've just basically made up this group between us, led by Rosie Schroeder. So Rosie actually organized the first New Zealand-Australian plant cell wall meeting in Auckland last year, which is where I met a lot of these wonderful people that I currently came to visit, like Deidre, Patricia, etc., and Mike as well, which is how I set up this collaboration and I'm currently working on. So in the cell wall group itself, there are a lot of scientists with a range of different backgrounds. So we have from microscopists to people who work in genomics, as well as molecular and biochemistry work as well. So I put over here a small map of Palmerston North, which is the city I actually live at now. It's actually quite small. The city only has 1 mall, unfortunately, and about 5 supermarkets or so. So this is literally the whole map. And I thought it was quite cute because in the rural areas, they've put little sheep logos over here to let you know, oh, it's rural. And it's a university town, so a lot of the population is actually made up of university students because there's Massey University across over here. And where we are, we're located in this little region. We're nestled in a complex together with Ag Research, Fonterra, and Assure Quality. So that's where we are. Just a few photos. There is also a river that runs through Palmerston North, but it's nothing like the Brisbane River. There is no city cat that runs on it. That's my dog, by the way. And because it's quite windy up there, we harness a lot of energy from windmills. And this is quite a typical photo. You see a lot of sheep up there as well. That's an overview of the city. And it's not really coastal, but there is a beach that's nearby as well. It's a West Coast beach, so it's quite lovely out there. Okay, I guess at some stage I have to talk to you about work. So basically what I'll be telling you today about is mostly on the breeding program that I work on potatoes. So at Plant&Food Research, we have an extensive potato germplasm collection and successful breeding programs that has seen the introduction of potato cultivars to the marketplace, such as the Golden Delight variety that you can find over here. That's a Plant&Food variety. So in the past, our breeding programs have largely focused around crop yield and pest and disease resistance. However, in the recent times, there's been a push towards breeding programs that can introduce new cultivars with novel traits. These novel traits include texture, and texture is mainly depicted by the end use of the potato. So whether it's to be a mashing variety or a roasting variety like that, they breed for that. Flavor, novel size, because you can breed for potatoes that can be longer to suit the French fry industry. Or also in recent times, there's been a push towards releasing potato cultivars that can offer health and nutrition benefits. For example, this purple variety over here, in New Zealand it's marketed as Purple Heart. I think in Australia it's known as Purple Congo. It's high in anthocyanins, so it's a healthy variety. It's marketed as being healthy. So the background of my research project was actually to screen our potato breeding program for cultivars that have low glycemic index. And this is because low glycemic index products are actually healthier for you because it can regulate blood sugar levels, and it's particularly important for people who suffer from type 2 diabetes. Also good if you're trying to manage your weight and satiety, things like that. So that was the focus of my project. So just to briefly take you through the outline of my presentation and briefly what I'll be talking to you about today. So we screen all the potato lines from our breeding program for their in vitro GI, and I used an in vitro digestion method for this because we were screening about 100 over cultivars, so the in vivo method wasn't really viable for us. And then I proceeded to do some cell wall analyses, and these are a list of the techniques that we've used, as well as some starch analyses. And because we don't have NMR facilities in Plant&Food Research, that's where the collaboration has come in with Mike Gidley's group. It's what I came here to work with Bernadine with. And so for today's presentation, I'll just be presenting results from these analyses. We have on the cell wall side the comprehensive microarray polymer profiling. Sometimes this technique is also known as glycan profiling. some immunofluorescence microscopy work, and some pectin analyses. And on the starch side, we have the amylose content analyses as well as granule morphology, and this was done using scanning electron microscopy. Okay, so why are we interested in studying the cell walls and understanding how this relates to the GI of the potato? This is an example of a scanning electron micrograph of a raw potato tuber. So you can see in its raw state, the cell walls actually act to contain the starch granules on the inside. So during processes such as cooking, chewing, and food preparation, the cell wall actually breaks. So starch then becomes accessible to your digestive enzymes to hydrolyze and release glucose. So the faster the rate of release of glucose into your blood, this corresponds to a high GI food, and vice versa. So what we were thinking is, could there be some cultivars that could have more stronger cell walls, that could be more resilient to cooking and chewing processes, that they could act to protect the starch a little bit more, and this would then restrict the accessibility of the digestive enzymes, thus resulting in a low GI potato. So this was sort of the background or the idea behind our project. Right, so just to briefly take you through the in vitro digestion method, this was based on the English method which we used. So we started out with two preparations, the whole potato up here, which we boiled to cook it, and then we passed through a mincer, and this was just to mimic the chewing procedure. In the other preparation, we isolated the starch first, and this was done by blending the potato tissue in water and allowing the starch to settle, and then we separate that. And so this preparation contains both the starch and the cell walls. This preparation contains only the starch without the cell walls. So they are then taken through what is a mock digestion procedure. So you have over here conditions that mimic the stomach. So we have hydrochloric acid and pepsin. And then down here, conditions that mimic the small intestine. So we have the two enzymes, amyloglucosidase, and the pancreatin, which contains the alpha amylase to digest the starch. So once these enzymes are added, what we do is we remove two aliquots, one at 20 minutes and one at 120 minutes. This is then quantified for glucose using a Goldpot calorimetric method. And the 20-minute aliquot corresponds to rapidly digested starch, whereas the 120 corresponds to the total available starch. So the in vitro GI is then calculated as a percentage of the rapidly digested starch out of the total available starch. So this gives us an index down here of what we use as the in vitro GI estimate. After this procedure, there is also an insoluble pellet that remains. So this is essentially what remains undigested. This is mainly where you'll find your dietary fiber or your cell walls. Okay, on to some results. So this is a truncated graph of all the lines, all the potato lines that were screened. In all the experiments, we use white bread as our positive control. And over here on this axis, we have the in vitro GI index. In the blue bars, we have the starch and cell wall preparation, and in the red bars, the starch-only preparation. And out of all the lines that we screened, most of them did not show a significant difference, but there were three lines which we identified, lines number 3, 4, and 8, indicated by the asterisks over here, whereby we saw that the starch and cell wall preparation had a significantly reduced GI compared to the starch-only digestion. So we wondered actually why this was and why these three lines were showing a different digestion behavior to the other potato lines. Could it be that somehow their cell walls are protecting the starch, so that's why this bar is lower than the red one over here? So we then went on to do some cell wall analyses to try and understand this. So this is the first cell wall analysis technique which I'll briefly describe to you. It's quite a new novel method. It was, the protocols were first developed in the Willard's group in University of Copenhagen. This method is known as the comprehensive microarray polymer profiling method. So basically what we do is we isolate the cell walls first from the potato tissue, and this is done using an ethanol acetone precipitation method. And then we fractionate the different cell wall components to release what we're after. So for example, in water, we get the freely soluble pectin. In CDTA, we can release the calcium chelated pectin. in sodium carbonate, we get the ester-bound pectins. And in the 4-molar KOH friction, we get the very strongly bound pectin as well as the hemicelluloses. So these are all components in the plant cell wall. After recovering the cell wall components, we plate them, and using this microarray robot, it spots it onto a nitrocellulose membrane. After that, the procedure from here on in quite resembles a Western blot or an ELISA, if you're familiar with those techniques. So we block for non-specific binding, and then we incubate with our primary monoclonal antibody of interest. At this stage, you can also use a carbohydrate binding module. And a lot of plant cell wall directed antibodies can be found from these sources over here. And then we choose an appropriate secondary antibody. And after that, the array is developed colorimetrically. And for this study, I've used NBT-BCIP. The arrays are then scanned, and then we proceed to analyze the data. And what's actually quantified is each spot's integral intensity. So I've put over here an example of what the array looks like. So instead of a Western where you get bands, over here we get spot development instead. And so we can actually quantify the intensity of each spot. And this is related to the abundance of that epitope in which the primary antibody was directed against. So the results can then be presented as heat maps as such. So in this example over here, I've selected 3 antibodies which are specific to 3 different regions on the homogalacturonine molecule of pectin. So we have LM19, which is an antibody that goes for the low methyl esterified regions of pectin. We have LM20 that goes for the highly methyl esterified regions of the pectin. And then we have 2F4 that goes for the calcium-bound pectin. So over here on the left, we have the cultivars which I've analyzed, and over here, the different fractions of the cell wall. So in this heat map, you can see that the epitope with highest abundance is LM20, which is the highly methyl esterified pectin over here, and it was most extractable in the water and CDTA fractions. The CDTA fraction also seems to contain relatively high proportions of all 3 of these epitopes of pectin. In this next slide, I've now selected 3 other antibodies. We have LM5 that goes for the galactan side chains of rhamnogalacturonin 1 on pectin. So this is a different molecule of pectin. LM6 that goes for the arabinan side chains, and we have LM13 that goes for the longer and more linearized arabinan chains instead. So over here in these results, we see that the galactan epitope occurs in highest abundance, and this is not very surprising because potato cell walls are known to contain high amounts of galactose. So this was something that we did expect. But what was very interesting for our study was over here in the 4-molar KOH fraction, we saw that our 3 potato lines of interest, lines number 3, 4, and 8, they showed a much higher abundance of all 3 of these epitopes compared to the other potato lines down here. So in this 4-molar KOH fraction, it solubilizes the pectin that is very strongly bound in the cell wall. So this was indicative to us that these 3 potato lines could actually have cell walls that could be stronger compared to other potato lines. Also in this study down here, they have identified that galactan and arabinan side chains are actually able to bind in vitro to cellulose microfibrils via very strong bonds such as hydrogen bonds. This actually would explain why we would require a strong alkali treatment to release these classes of the epitopes down here. So this was sort of just very interesting for us because it pointed out that these 3 cultivars actually did show a difference in the pectin classes, especially those that are solubilized in the 4-molar KOH fraction compared to other potato lines. Right, so after that, what I also did was after the 4-molar KOH treatment, the residue that remains is cellulose rich, so we can also quantify the amount of cellulose that's present in there. So just to see if they were any different in our 3 potato lines. Over here we have in the gray bars the amount of cellulose in milligrams of dry cell wall basis, and then also taking into account that the different potato lines can have different amounts of cell wall itself. We then also calculated it on a fresh weight tuber basis in the green bars. So you see that the three lines, lines 3, 4, and 8, are not collectively as a group any different from the other potato lines that were analyzed. However, if you look at the green bars, there is a variation in cellulose content that can exist between the different potato lines. And this is merely reflective of the different dietary fiber content that you can have in different potato cultivars. Right, so because our glycan profiling results seem to indicate that there was a difference in pectin, what we then also did was try to quantify the actual difference itself in terms of amount of pectin. So I quantified the pectin in the raw tuber, and this was just using a colorimetric galacturonic acid assay. And also the amount of pectin in this fraction down here, what we have termed the surviving pectin, because this is the pectin that remains in this insoluble pellet that is able to survive the cooking as well as the in vitro digestion procedure. So this would be the pectin that is most resilient. So the hypothesis here was that if the cell walls in those three potato lines, lines 3, 4, and 8, were stronger, they could also possibly have more pectin content coming out on this end. So here are the results. We have in panel A the amount of pectin in the surviving pectin fraction, and in panel B pectin loss calculated as the pectin content in the raw tuber subtract the surviving pectin amount. So in panel A we saw no significant difference in our three lines, which are the first three on the left over here, compared to the other potato lines that were screened. However, when we looked at it on a loss basis, they were showing significantly lower pectin loss compared to the other potato lines. So this just shows that these three lines are losing less pectin throughout the cooking as well as the in vitro digestion procedure. So this was also interesting for us. What we then also went on to do was some immunofluorescence microscopy work, because while it's all well and good to analyze the amount of pectin, this and that, we actually also wanted to visualize where in the cell wall these pectin epitopes were occurring, and if they were spatially distributed in a different way across the different cultivars, because this could have an impact on the properties of the cell walls itself. So over here I have sections of the potato. These are raw potato sections that have been resin embedded in section and then stained using those same monoclonal antibodies. So we have in this example LM5 that goes for the galactan side chains. On the left we have one of the low GI lines that we're interested in, and on the right we have a high GI control line. So for LM5 labeling in the walls, they don't label the middle lamella, which you can see is the region between the two cell walls over here. They also don't seem to label the junctions. They only label sort of the inside of the walls over here. When you compare immunofluorescence microscopy images, they are only qualitative, so they don't give you any quantitative information. So this is actually purely for us to visualize where the epitope is occurring within the wall. So in the two lines, we didn't see any difference between the two for labeling. But when we had a look at LM19, this is an antibody that goes for the low methyl esterified region of pectin. We saw that most of the low GI lines actually displayed this labeling pattern where it was strongly labeled throughout the primary wall and very intensely in the middle lamellar zones. Whereas some of the high GI cultivars, it only preferentially labels the corners of the walls and the tricellular junction zones, which are these zones here where the cells actually meet. In the literature, these are also the areas of the cells which are very abundant in calcium. So I've put over here two sets of images in low mag and high mag to better illustrate this difference. So you can see over here, it labels intensely in the middle lamella areas, and over here it seems to only really like the corners. So what this actually showed us is we're not actually presenting this and saying that, oh, all low GI lines label this way and all high GI lines label this way. That's not true at all. What we actually saw was that the same epitope can have a difference for preference of location within the wall, so that they're not all the same. not all the potato cultivars are the same. And why some actually have more in this region and some only have some in this region is something that's not well studied in the literature, and it's not well known at all why this happens. So what we're trying to better understand over here is how this can actually affect the functionality of the wall and how this affects the low or high GI behavior of these cultivars. So onto the second part, we also could not ignore the fact that the potato contains a lot of starch. So we also had to concentrate a little bit on starch analyses, which for today's presentation, I'll just present these two techniques with you. So when we had a look at the amylose content, this was just done using an iodine binding method. In the literature for cereal starches, the in vitro GI and amylose content actually has a negative correlation. So we wanted to understand whether in our potato samples that we were screening, whether this was also true. So we had a look, and the red dots are the high in vitro GI lines and the blue dots the lower ones. And our 3 lines of interest are over here. So they were not actually grouped together to be any different from the other lines. However, we did see that most of the high GI lines tended to be on the lower amylose content side, and most of the high GI were over here. Also had a look at some starch granule morphology. So this was done using SEM. The problem with doing a lot of microscopy work with potatoes is that we only can look at the potato in its raw state, because once it's cooked, the starch actually swells and gelatinizes, and it just overwhelms everything. And so all you will see is just like a gray mass covering everything. So we can't actually see any of the cell walls and stuff like that we're interested in. So we can only have a look at it in its raw state. And so on the left, again, we have one of the low GI lines that we're interested in, and on the right, the high. So initially, when we set out to do this work, we were quantifying to see if the starch granules actually have a difference in average size. However, when I began this work, I quickly came to realize that it was quite difficult, because there seem to be 2 very distinct populations of starch granules. You get some that are very small, that are almost under 10 microns, or you can get some that are really big, some up to like 80 or closer to 100 microns. So therefore, there was a huge range in the size of the starch granules, which seemed to indicate to us that if we were looking for an average size, this would not give us an accurate representative answer to what we were looking for, because the range being so huge. So what we then went on to do was we thought, okay, maybe we should quantify the size and do a distribution plot instead, so we could actually see if there were more small ones or more big ones. But there wasn't any significant difference. Also, this turned out to be very, very time consuming, because any of you that do any microscopy work, you might know that we have to look at multiple images to get a good representative overview, especially if you're looking at cultivar differences. You have to section multiple sections of the same tuber and then the same cultivar many times to actually get a representative answer. And so for us, this was also not a viable option, because it was hugely time consuming. Which is why I've actually set up this collaboration looking at the other starch properties in terms of its crystallinity index and amylopectin branching using NMR techniques, which Mike's group has expertise in. Okay, so just to wrap up over here, in our work we have identified 3 potato lines, lines number 3, 4, and 8, that have a lower in vitro GI that seems to be affected by the presence of their cell walls. So from our glycan profiling results, their cell walls seem to contain pectins that could be more strongly associated with cellulose. They also show less pectin loss during cooking and in vitro digestion procedures. And currently, because Mike's group has really good expertise in starch analyses, we're still doing the ongoing starch work. So I'd just like to conclude and thank the people who have been involved in this project, John Monroe and Mishra for the digestion work, and Arian and Day for the cell wall side of things, and Isabel Muller, who helped us with the glycan profiling setup in our lab. And John and Russell are the potato breeders that I work with, and AgMut for funding this research. Thank you.